Chapter 10 is looking at how genes work, and we're going to be looking at the story from the textbook about tobacco and how we're using tobacco to create or increase the amount of flu vaccines that we have available, and growing these tobacco plants that create the proteins for the necessary vaccines. It will increase the amount of vaccines that we can produce, and it will also increase how fast we can produce these vaccines. So if you remember from the previous chapters, we were talking about these two terms, genotype and phenotype. So if we remember that differences in genotype, which is the DNA nucleotide sequence, this is going to be expressed, or it will create differences in the phenotype. And remember, the phenotype are those traits or characteristics or those physical properties of an individual. These physical properties are created by proteins. So what we're going to be looking at and kind of relating this together is how this DNA is expressed into proteins. So this chapter, we're really going to focus in on this blue arrow right here, linking the genotype to the phenotype. So in the story in this chapter, they talk about growing tobacco to help treat the flu or to make flu vaccines. So what they're talking about is that we are growing tobacco plants to make the components that are put into the flu vaccines to help protect people against influenza. And kind of the important part, or why they're using tobacco, or why they're actually looking at this, that traditionally, for most vaccines, we grow the particles or the proteins for the vaccines in chicken eggs. But now they're investigating other ways to produce the products needed for these vaccines, specifically looking at tobacco plants. And this kind of summarizes why researchers are switching from egg production of vaccines to a plant production. So in order to make a vaccine, we need a large amount of pathogen proteins to put into the vaccine. These vaccines will then inject into people, which will trigger our immune response. So we talked about that way back in one of the earlier chapters, but making those vaccines is really important because our population is growing very quickly and more people want access to these vaccines. So we actually have discovered that so many people are getting the flu vaccine, but in previous years we have actually run out of flu vaccines, or people would have to wait in order to get it, which can put everybody at risk for complications from influenza. So in order to create these pathogen proteins needed to put into the vaccines, this company, they're going to, instead of using eggs, they will use plants. And two things that are really important is that using the tobacco plants, it causes the pathogen protein to be created 6 times faster. It's also more cheap compared to growing these protein pathogens in eggs. So just to kind of compare, so here we have tobacco plants and some things about it, and then the traditional method of producing these pathogen proteins in eggs. So one I mentioned is the speed. So in tobacco plants, we can produce vaccines within a month. Eggs, it takes 6 months. The cost, $36 million versus $400 million. Another big thing with vaccines is that some people are allergic to eggs, and if you're allergic to eggs, you cannot receive vaccines. So this would cut down on any allergy risks because tobacco plants have a minimal allergy risk, whereas individuals that have egg allergies, they can't get the current vaccines. And then our availability, these tobacco-grown pathogen proteins, they're not yet approved by the FDA, whereas the ones in the eggs have been approved. So that is one thing that is kind of stopping us from producing vaccines using these tobacco plants. So this idea of using organisms to create a product is referred to as biofarming, specifically manufacturing these vaccine proteins in plants. So bio, remember, refers to life, and then farming, this is a play on farming with an F, and pharmaceutical, like pharmaceutical drugs. So biofarming is using different organisms in order to create pharmaceutical drugs. And the example here, getting those vaccine proteins being created in these plants. So how this is done is through a process called gene expression. So in our tobacco plants, we have to have the gene to create those proteins for the pathogen. That tobacco plant will then express those genes to create the proteins. So this is a process by which genes are transcribed. So this is the first step into an RNA molecule. And then that RNA molecule is translated to make the proteins. So this is what we're going to be looking at within this chapter, is this gene expression, this transcription, and translation. So we're going to look at an overview first, and then we'll get into the details. So first, transcription. This is the first step of gene expression, and it's when information in the DNA is converted to information in RNA. This happens in the nucleus of our eukaryotic cell. And it's diagrammed here, I just circled it. The other part is the translation. This is the second step of gene expression. This is when that RNA that was just created in the transcription step is read and converted to give us what order of amino acids we should put together to create the protein. And remember that proteins do pretty much everything in the cell, so these proteins will give all of those different traits or characteristics that an organism can have. So it can code for skin color, hair color, can code for potentially how tall you're going to grow. It can code for those pathogen proteins that we need to put into the vaccine. So these two terms, or these two steps, transcription, translation, they both kind of start with the same letters, so it's kind of easy to get these two steps mixed up. So I'm going to go over a quick way just to remember transcription versus translation. The word transcription means to copy. So when you transcribe something, you're just making copies of it. And this is exactly what is happening during the step. So what's happening is we're taking that genetic information in the DNA, and DNA stands for deoxyribonucleic acid, so it's a nucleic acid. We're using that to create RNA, which is ribonucleic acid. So we're just copying that genetic information from one nucleic acid form into a second nucleic acid form. That RNA molecule is then going to go through translation. And to translate something, it just means to go from one language to another language. In this instance, we are going to translate from a nucleic acid language into a protein language. So kind of think of it as translating from English to Spanish. So we're going from a nucleic acid type of molecule made up of nucleotides into a protein type of language made up of amino acids. So I kind of hope that helps you to keep these two different terms separated from each other. So we're going to look at transcription first. So we're going to look at how our DNA information is copied to create an RNA molecule. So this transcription, we're going to synthesize an RNA based on a DNA template. And transcription requires a couple of things. So on that DNA template, that is going to be called our gene. So if we look at a sequence of DNA up here, so I drew a gene. On that gene, we're going to have a sequence of DNA near the beginning, and that's called the promoter. This promoter is going to tell the next part where it should actually start the transcription process. The molecule that does the transcription process is called RNA polymerase. And this is an enzyme that will read that DNA template and synthesize that RNA molecule. So this RNA polymerase, it's going to attach on to that promoter. It's going to travel down the gene and create an RNA molecule. At the end of the gene, there is a sequence called the terminator. So the terminator, this is basically the end of the gene. It tells that RNA polymerase where it is going to stop this transcription process. So what this looks like, so our RNA polymerase is going to attach on to the promoter or the start of the gene. That RNA polymerase is going to unzip that DNA double helix and prepare to travel down the gene and create the RNA molecule. The RNA polymerase is going to read the sequence of DNA template nucleotides as it travels down the gene. And as it reads that DNA template, it's going to insert a complementary RNA nucleotide. And this is going to construct a messenger RNA or mRNA molecule. That DNA polymerase is going to continue to travel down until it reaches the terminator or the end of the gene. So this is a pretty straightforward process. RNA polymerase attaches on at the promoter, reads that DNA template, creating the RNA molecule. Once it reaches the terminator, it's going to stop. This RNA molecule that is being created, it is created using complementary base pairing rules. So this hopefully sounds a little bit familiar. So our DNA, when our DNA double helix is put together, we have complementary base pairing rules where adenine goes across from thymine, or A's are across from T's, and guanines are across from cytosines, or G's are across from C's. So a similar thing happens when we go from DNA to RNA, although there is one little twist. So these are those complementary base pairing rules. And the big difference that we see is the first one. If your DNA molecule has an A, RNA will put a uracil. G goes to C, C goes to G, and T's go to A's. So I'll show you an example of this. So if we have a DNA molecule, so here we have a DNA molecule A, T, C, G, G, T, A, C. Our RNA polymerase, when it creates the RNA, it's going to use these complementary base pairing rules. So if our DNA has an A, our RNA will have U. T will pair up with A. C will pair up with G. G pairs up with C's. T pairs up with A. A pairs up with U. C pairs up with G. So this again is that process where we take that DNA molecule and use it to create a RNA molecule. So again, that's our transcription process. These RNA molecules in eukaryotic cells go through something called processing or modifications. So in eukaryotic cells, but not prokaryotic cells, these mRNAs that were just created through the transcription, they undergo modifications before they can leave the nucleus. And there's a couple of things that happen. So one is that there's chemical modifications to both ends of that mRNA molecule that was just created. The second thing that happens is RNA splicing. So in this RNA molecule, there are non-coding regions called introns. These non-coding introns are cut out. The protein has coding regions called exons. These exons are kept and they are pasted back together. So I will show you what this RNA splicing looks like. So here we have our DNA molecule up here at the top in the blue. This DNA molecule will go through transcription. We use it to create an RNA molecule. Some of these regions, in this case, the dark red or dark orange regions, these are the introns. They are going to be spliced out or they are removed. The remaining exons, so the lighter colored parts, are kept. They are pasted back together to create the mature mRNA molecule. So this happens in the cytoplasm. It allows the cell to kind of clean up the information or those directions in that RNA molecule. Once that RNA molecule goes through processing, it goes into the second step, which is translation. So translation, this is a process by which ribosomes, they convert the information in that mRNA molecule into proteins. So this requires a couple of different things. So we have our template mRNA that we just created in our transcription. We have a cell structure called the ribosome. And we talked about this when we talked about cell structure. Ribosomes are responsible for protein synthesis. So these ribosomes are made up of an rRNA molecule and proteins. So what they're going to do is they're going to read the mRNA that was made in transcription. They'll read it in a set of 3 bases called a codon. That codon will then match up with what's called an anticodon on another type of RNA called tRNA. This is transfer RNA, so it has an anticodon on it that matches up with the codon. And then on the other end of the tRNA, it has an amino acid attached to it. And remember, amino acids, these are the building blocks or the monomers for proteins. So how this works, this mRNA molecule is transported out of the nucleus into the cytoplasm, and it's going to be attached to a ribosome. The ribosome is then going to read those mRNA codons, those sets of 3 nucleotides. It's going to set up a complementary base pairing with anticodons on the tRNA. This tRNA then on the other side has the correct amino acid attached to it that matches up with the codon on the mRNA. These amino acids, when they're brought in one by one, they're going to be linked together with covalent bonds to start forming a protein. So what this looks like, so here we have our ribosome, which are the purple. There's 2 purple subunits. We have our mRNA molecule that's threaded through the ribosome. This mRNA molecule, we read it in codons. So the first codon would be AUG. This codon matches up with an anticodon on the tRNA. So that would be UAC is our anticodon. On the other end of the tRNA, we have our first amino acid up at the top. So then what happens is the ribosome is going to shift down, read the next codon, which in this case will be CUG. It brings in a tRNA with the anticodon, that's complementary, which would be GAC. On the other end of the tRNA, we have our next amino acid attached right here. So the second tRNA comes in, our codon and anticodon match up with each other, they're complementary. We bring in our second amino acid. Our first amino acid then covalently bonds to the second amino acid. This tRNA on the left-hand side is then going to exit out because it's no longer needed. The ribosome will shift over another codon, or 3 nucleotides to the right. It will read that third codon, bring in the third anticodon with the third amino acid. That enters in, and this whole process will repeat over and over and over. So we'll build up this protein 1 amino acid at a time by reading 1 codon on our mRNA at a time. So in order to bring in the correct amino acid when having the codon match up with the anticodon, we have a chart called the genetic code. So this genetic code is unambiguous, it is universal, and it is also redundant. So these things are very important. So basically what this states is that there are 64 different possible codons. 3 of them are used as stop codons, so they don't actually code for an amino acid. So there's 61 codons that code for amino acids, and there are 20 possible amino acids. So this is the redundant part, so there can be 2 to 4 actually different codons that code for the same amino acid. But every codon only codes for 1 amino acid. This genetic code is also universal, so it is pretty much identical in bacteria as it is in grass, as it is in humans. So every single living thing follows pretty much the same identical genetic code. So it's a pretty neat thing to understand. And they have a nice little chart that you can use in order to read the codons and figure out which amino acid is going to be next. In addition, there is the start codon. So this is where this translation is always going to start with a codon AUG. And like I mentioned, there are 3 stop codons. So that tells the ribosome where it should stop doing translation. Then every other codon codes for a specific amino acid. So just to give you an example of how to use this chart, if we have the codon AAC, for example. So this is our codon AAC. The first letter in our codon is A. So we go over here to the left-hand side. It says first letter codon. We have A. So we're going to be in this row. So if you look across this row, every single codon starts with A. Our second letter is A. So this is across the top. We have U, C, A, G. We have A. So we're ending up in this box right here. And we specifically have AAC. So here we have AAC. So this is going to code for the amino acid asparagine. Right there. So it's very easy to use. They set it up. All 64 codons are listed, and it tells you specifically which amino acid it will bring in. So this is how our genes, our DNA, can be used to create proteins. And then these proteins will give us all of our physical traits or physical characteristics. So if you remember, our DNA is made up of sequence called genes. Those genes are transcribed, so they go through transcription to create a mRNA molecule. This mRNA molecule is then going to be read in codons, so this is the translation part, to figure out which amino acids are going to be hooked together to create that functional protein, which again gives us all of our traits and characteristics. So differences in those genotypes or in that nucleotide sequence within our DNA is going to give us or result in differences in that phenotype, specifically differences in the protein that is built, which will lead to differences in those physical properties, so those physical traits and characteristics. So the big key here are these differences. So differences in the genotype can be caused by mutations. And we talked about how we can have mutations during DNA replication. You can also have mutations during that translation or transcription. So DNA sequence that is mutated, it may not be able to be translated or transcribed. We could end up with an amino acid chain that ends prematurely, so it doesn't get fully formed during the translation process. Or if we do end up with a final protein, it can fold into its 3D shape incorrectly. So an example of this, we can have base substitutions in our DNA sequence. This will not cause something called a frame shift mutation. Where mutations where we have a base that is inserted or deleted, these do cause a frame shift mutation where the codons are shifted either to the left or the right one base pair. So these are different ways where we can end up with changes in that DNA sequence will create changes in the mRNA. Those changes in the mRNA will then create changes in which amino acids are put together. You either have the wrong amino acid put in, or it will completely shift the size or the length of that protein or that amino acid chain. So these genes that are being transcribed and translated, they can be regulated. Specifically, you can turn genes on or off. That's one type of regulation. So this allows the production of different proteins as needed by different cell types. So like I mentioned, genes can be turned on or off. We can upregulate gene expression, so we can increase or speed up that expression. You can also downregulate gene expression, and this is where you will decrease or slow down that expression. Gene expression can be controlled at several points.